Hello, everyone, and welcome to the Pathogen Prodigies podcast. This episode is hosted by Amelia, Diana, Luke, and Emily. This first episode of our podcast is on bovine respiratory disease. To begin, we'll start off with what is bovine respiratory disease. Bovine respiratory disease, also known as BRD, is a multifactorial disease complex that causes bronchopneumonia or pleuropneumonia in cattle. Bovine respiratory disease affects cattle in both the beef and the dairy industries, but typically affects calves.
The bacterial pathogens that are part of this BRD complex include Pasteurella maltosida, Mannheimia hemolytica, Histophilus somni, and Mycoplasmopsis bovis, also known as Mycoplasma bovis until recently. P. maltosida, M. hemolytica, and H. somni are all members of the order Pasteurellales and family Pasteurella M. bovis is in the order Mycoplasmatales and the family Mycoplasmataceae. Some other less common bacterial pathogens include Bibbersinia traehelosi and Truparella pyogenes. These bacterial pathogens have also been implicated in other diseases as well, and some are involved in respiratory diseases in other animals.
In some BRD cases, a primary viral infection precedes a bacterial infection, weakening the host immune system, allowing for easier colonization. Bovine respiratory syncytial virus, or BRSV, is scientifically known as orthopneumovirus bovis. BRSV infects the upper and lower respiratory tract and is spread by nasal secretions. This virus is closely related to human respiratory syncytial virus, HRSV, and can be used interchangeably for research purposes. Symptoms include increased respiratory rates, anorexia, serious nasal discharge, dry muzzle, and malaise. BRSV survives because of its heterogeneity of its genome and low fidelity and replication. Bovine viral diarrheal virus, BVDV, is a pestivirus from the family Flaviviridae. BVDV has high morbidity and mortality rates, increased premature culling, and decreased reproductive performance. When infected with BVDV, cattle are often asymptomatic, but multiple body systems can be affected, including the reproductive, respiratory, gastrointestinal, circulatory, immune lymphatic, musculoskeletal, integumentary, and central nervous systems. Cattle can be infected with BVDV by physical contact between infected cows, vertical transmission, and indirect exposure to secretions or excretions containing the virus. Bovine herpes virus 1 causes the following symptoms: significant inflammation and damage of the upper respiratory tract characterized by rhinitis, laryngitis, and tracheitis. Other bovine viruses have been less commonly implicated as a primary viral infection in BRD. Since the infection of these viruses can lead to a weakened immune system in fighting the implicated bacterial pathogens, it is recommended to vaccinate against these viruses to aid in the protection against cattle developing BRD.
Now that we know the pathogens in play, what is the life cycle of BRD and how is it transmitted? BRD develops as a result of a complex interaction of various factors. These factors include environmental factors, host factors, and pathogens. Environmental factors such as crowding, weather, dust, transport, weaning, and ventilation issues can serve as stressors that can affect the host's immune system or even increase transmission rate. Viruses can also create openings for bacterial infection in two different ways. First, a virus can damage respiratory clearance mechanisms, causing the translocation of bacteria from the upper respiratory tract to the lower respiratory tract in order to colonize. Another tactic used by viruses to pave the way for bacterial infection is to suppress the immune system response entirely. Due to a complex interaction of numerous factors, bacterial pathogens infect the lower respiratory tract to cause bronchopneumonia. The complexity of BRD's life cycle makes it incredibly difficult to homogenize a diagnosis for every case. The transmission of the BRD is complex. M. hemolytica, P. maltosida, H. somni, and M. bovis are all considered normal flora of the nasopharynx, and therefore does not always lead to infection. These bacteria often become opportunistic pathogens and infect the lower respiratory tract. BRD's notoriously difficult life cycle and transmission patterns are the reasons the organism is able to ravage cattle herds.
Now, even though BRD's transmission can be complicated, BRD's impact on cattle in terms of morbidity, mortality, and reproduction is incredibly clear. BRD is a common cause of death in cattle, and it can range in its severity. Clinical signs include nasal discharge, fever, heavy breathing, and anorexia, which can lead to pneumonia and respiratory distress if not treated promptly. Additionally, BRD can affect the reproduction of infected cattle, reducing fertility and leading to fewer healthy offspring, which can have long-term effects on the health and productivity of cattle herds. This not only causes suffering for the animals, but also results in significant financial losses for farmers and those who depend on their production.
Thankfully, there's an acronym for farmers to use when they suspect their animals are infected with BRD. Livestock owners are supposed to observe D for depression, A for appetite loss, R for respiratory changes, and T for temperature changes, or DART for short. Further techniques by healthcare professionals can be done to detect BRD in both antemortem and postmortem scenarios. Bacterial culturing or tissue sampling can be done to find active cultures in cow herds, whereas transachial washes and nasal swabbing can be done to postmortemly identify the cause of infection.
There are even some preventative measures cattle owners can take to ensure the safety of their livestock. Vaccinations exist that can provide immunity to an entire herd of cattle for the primary viral pathogens. Vaccinating cattle for these viruses can help limit the potential development of BRD, as these vaccinations will help maintain an intact immune system. In the treatment of fever, veterinarians may prescribe non-steroidal anti-inflammatory drugs. However, this does not treat any of the other symptoms, not to mention all of the antimicrobial treatments available to treat any minor cases. Typically, the antimicrobials used to treat for a BRD infection are long-acting antimicrobials. Of these antimicrobials, macrolides such as fluorophenical are typically used in pre-weaned and weaned cattle, while beta-lactams such as third-generation cephalosporins are typically used in adult cattle to treat bovine respiratory disease. Other antimicrobials used include fluoroquinolones, tetracyclines, and sulfonamides. Recently, the Food and Drug Administration approved the usage of third-generation fluoroquinolone called protofloxacin to treat bovine respiratory disease. However, as you probably expected, antimicrobial resistance can contribute to the difficulty of disease treatment. In a study based on the trends in antimicrobial resistance over time, it was shown that the level of resistance to an antimicrobial is dependent on the pathogen, in which some of the pathogens show more resistance than others.
All the bacterial pathogens can be cultured using general lab culturing methods. These can also be identified through genetic testing and serological testing. The primary viral pathogens can be identified through genetic or antibody testing depending on the circumstances. Currently, the most common way to study bovine respiratory disease, as described by Gandhi et al. In 2023, includes submerged tissue culture and air-liquid interface. Submerged tissue culture consists of epithelial cell monolayers that are typically overlaid with cellular or spent biofilm culture medium. Air-liquid interface involves the exposed apical side of epithelial cells that are allowed to differentiate. To study BRD, three-dimensional bovine models are used to represent the intricate details of respiratory tract using multiple cell types.
This disease is very important economically in the beef and dairy industries. Bovine respiratory disease is the costliest disease affecting the cattle industry, persisting despite efforts to reduce the disease and its resulting economic losses. It is estimated the cost of BRD in the United States is around $1 to $3 billion. As for the mortality cost, the total value of non-predator death loss in cattle and calves was reported as $3.6 billion, with $907.8 million, or 24.6%, because of the respiratory disease. This data reinforces just how much of an impact this disease has both on the economy and agricultural production.
In terms of commonality of the disease, nearly 20% of all cattle raised for beef production will require clinical treatment for BRD at some point in their lives. BRD is the second leading cause of death after digestive diseases. Bovine respiratory disease is one of the most extensively studied diseases in cattle. However, finding the exact number of citations per year has been difficult to find. The primary viral pathogens, BVDV and BRSV, as well as bovine herpes virus 1, that predispose cattle to bacterial pathogens, M. hemolytica, P. multocida, H. somni, and M. bovis, which cause bovine respiratory disease, all have sequences in the GenBank database. Research into this disease began in the late 1800s and continues today. Since this disease is multifactorial and the bacterial pathogens are all normal flora of the nasopharynx, it is difficult to pinpoint when and where it was first reported.
While conducting research on this disease, we were unable to find any information on if the pathogens implicated in bovine respiratory disease can get diseases themselves. This could be an interesting avenue of future research.
In 2020, a case study out of the state of Texas exhibited a well-known, albeit small problem had transformed into a concerning trend. Over the past several years, a steady increase in cattle testing positive for bovine respiratory disease had become apparent. This problem had been rigorously studied for generations, but in the past few years, the disease has become acute. Despite having a normal appetite, large pastures, and healthcare services available, these cows were gradually losing bodily condition. The most notable symptoms were chronic watery diarrhea and frequent nasal discharge, although it wasn't always consistent. The true culprit behind this particular case was called the bovine viral diarrhea virus, or BVDV. This virus is actually a subset of the bovine respiratory disease, but still falls under the same concerning trend.
The most common hypothesis for the cause of infection is that the virus snuck into the animals' biological systems within the first 6 months of their lives before laying low. It is believed that the virus remained dormant, hiding for months to years before making its presence known. Severe sickness, widespread outbreaks, and even fatalities can begin as soon as the virus reactivates. Thankfully, faculty at the University of Texas A&M testing for BVDV was possible. Several standard options could be used, such as analyzing a serum sample for antibodies to the virus. Other techniques available for identification include search for the virus itself in the cattle's feces, or even using PCR to narrow down specific candidates.
Despite the effective identification methods, there was a catch. Researchers realized that the disease took a significantly long time to show itself. This means that even constant screening of young and adolescent cattle may lead to false diagnoses. The long dormant period of the virus is detrimental to any pre-screening trials that may have warned professionals. And if that wasn't enough, vaccination for BVDV was initially unavailable for the virus. The only way to control it was to remove positive animals from the herd and try to limit the exposure of young calves to the organism. Only very recently have live attenuated vaccines been shown to be effective against BVDV. This case study highlights just one small area in southern Texas that was ravaged by this disease. In the continental United States, outbreaks can occur in several states at once, raising mass concern.
To conclude this episode, we would like to dive into what isn't known about bovine respiratory disease and what would be some interesting areas for more research. One area of potential research could be in the true impact of certain environmental factors. The impacts of shipping cattle after their sale on the development of BRD could, with more research, help those in the cattle industries make decisions that aid in the health of their animals. Aspects that may impact the effective travel could be fasting and dehydration. Weather changes and interplay of climate variables could affect instances of BRD, and with more research showing the impact, veterinarians could use this knowledge to influence their diagnosis process. In regards to sale of cattle, it is generally thought that buying directly from a farm or ranch limits predisposition to BRD as compared to buying through sale barns, since in sale barns there are many new young animals housed together. More research into this to develop a more concrete connection of sale barns to predisposition of BRD could influence the structure of buying and selling cattle to limit potential transmission of BRD and maybe other diseases.
Another area of a bit more research is the impact of animal weight on predisposition to BRD. It has been shown that animals weighing less than the average weight are 1.4 times more likely to develop BRD. Further research to support this claim could provide farmers important context to how feeding can impact their animals. Currently, there are surprising yet conflicting studies into BRD development and sex, with multiple surveys finding it more prevalent in male calves and other studies finding it more prevalent in heifers. We believe that conclusive evidence on this statistic would not only answer the question of how sex impacts susceptibility to BRD, but also provide more information about the disease itself in general. In cattle, body alterations such as castration or dehorning could affect the immune system of cattle, specifically the age of these procedures. More research into what effect this has could influence farmers to make different decisions on not only what cattle to breed, sell, or buy, but also the time that they elect to have these procedures done for their cattle.
Clearly, BRD is an exceptionally relevant and dangerous disease.
Yet, despite the countless endeavors to try and understand it better, there are still gaps in our knowledge pertaining to the disease. For example, scientists currently believe that heredity of BRD is susceptibly low. There have been few studies looking indirectly into the breed effects on the susceptibility of developing BRD. To the species level, it has been shown that Bos taurus is at a greater risk of BRD compared to Bos indicus, likely due evolutionarily to the climate factors. Bos indicus originates from warmer climates. Further research into both breed and species factors could impact breeding patterns in both the dairy and beef industries. Yet, there is no way to pinpoint a baseline level of susceptibility due to the wide variety of each case in terms of breed of cattle, environmental factors, and strain of bacteria. Further metabolic tests could also be done to identify mechanisms used by BRD. Many speculations can be drawn by researchers using parental and genetic variation information. One such observation shows calves from younger dams have a higher pre-weaning BRD, but lower post-weaning BRD. This could be attributed to a multitude of factors, but it's possible that resistant dams had calves that were more susceptible to BRD. Along the same vein, part of a calf's acquired immune system relies on the mother's antibody level in the milk it provides for its offspring. Older mothers may have strong immune systems and can therefore provide their offspring with more antibodies. This may be a delicate balance, however, as mothers that are too old may have the opposite effect.
To summarize what we have discussed today, bovine respiratory disease is a multifactorial disease complex that is important for both the beef and dairy industries. BRD is an incredibly complex interplay of environmental factors, host factors, and pathogens. Primary viral infections such as BRSV, BVDV, and bovine herpes virus 1 can predispose a bacterial infection. This bacterial infection can be caused by any of the following, such as M. hemolytica, P. multocida, H. somni, and M. bovis, which are all normal flora of the nasopharynx. Colonization of the lower respiratory tract of these bacterial pathogens can cause broncho or pleural pneumonia. Prompt diagnosis and treatment can help limit the mortality of this disease. However, it can be difficult to treat due to the complex interaction of multiple factors that cause BRD. While there are some aspects of the disease that we still do not know, there is still ongoing research into bovine respiratory disease that will hopefully provide new knowledge that can help limit its impact on the dairy and beef industries. Thank you for tuning into our podcast on bovine respiratory disease. And from all of us at Pathogen Prodigies, thank you for listening.